Lucio Gama, who I first met when he was collaborating and working with Janice Clements here at Johns Hopkins. And since then he still maintains contact here as an associate professor in comparative molecular biology, but has moved on to bigger and greater things as the scientific director for collaborations at the Vaccine Research Center of the National Institutes of Health within the NIAID. And within there, he is responsible for the assessment, tracking, and coordination of all collaborative efforts between the Vaccine Research Center and extramural investigators, evaluating HIV broadly neutralizing antibodies, which in my opinion is the strongest candidate for a vaccine that has been developed to date. Still some ways to go, but it's very promising. In addition to work that he does here with latency and HIV infection of the central nervous system. So looking back, he was educated at the University of Sao Paulo, the largest city of Brazil. I know I pronounced that horribly, but I tried.
It's okay.
And almost his entire career has been spent on studying HIV infection in the rhesus macaque model. And in particular, he's been looking into seeding of reservoirs, and especially seeding of the brain reservoir back in the, I guess, late 2000s, early mid 2000s, when it really wasn't popular. There was a lot of pushback from the field at that time, whether or not macrophages could be infected, whether or not microglia could be infected, and it didn't matter, right? It didn't matter that these cells were infected. So certainly that's something that has changed over the years, thanks to the continued efforts of people like Lucio and certainly others. And in the last several years, he's put a great deal of effort into understanding the immune system and how the immune system reacts to HIV under conditions of suppression and under conditions when they're not suppressed, and then targeted broadly neutralizing antibodies in the SIV model as a curative strategy. So he's going to talk to us today about those efforts. Welcome.
Thank you so much. I'm going to start sharing my screen. Can you see it? Okay. So thank you so much, Norm, for your very kind introduction. As Norm said, I am still part of Hopkins. I'm a part-time associate professor at the Molecular Comparative Pathobiology department. But since 2016, I started working for the VRC. They needed somebody who knew the cure field and to start organizing all those trials that we are collaborating with, or for extramural collaborators that are using our products for clinical trials. In the beginning, I started only with bNAbs. Now I do bNAbs and HIV vaccines. The VRC has several HIV vaccines that are being tried. And I think that in the end, they're going to give me other pathogens as well. But at this point, I'm just with HIV. But I would like to start the talk today just with a piece of art from Frank Moore. He was a patient, or an AIDS patient in the 90s and 2000s, and he died in 2002. And I think it's very interesting, this painting that he did in, I think it was late 90s, where he described, through the painting, how he felt as a patient, right there in the middle of the circle and around the doctors and the social, turmoil that was happening at that time before you had art. But even there, you can see that we had a lot of conflict between between what we know, what we don't know. We saw that with COVID as well. But in terms of HIV, we still think, at least the population think that the epidemic is over. We know that now because of art and because of Truvada, a lot of people think that, it's controlled, but it's not. We know that 2020, since 1981, we had 70 million people HIV positive, 35 million deaths, 37 million people have HIV now, and among them, almost 2 million children. So we have still a long way to go. We still have a lot of people getting infected, especially in less privileged parts of the society. And although we have the tools to do that, We still do not know how this is going to pan out in the next, I don't know, 7 years. I know that Fauci said that by 2030, we're going to be controlling the HIV pandemic. Let's see if it's going to happen. But can we really end it? We already have the tools. And if you think about art is a wonderful combination that is able to control the replication. We know that once we start giving art to our patients, we have a decrease in death, a decrease in morbidity. We saw like increase in quality of life and the long lives for those patients. And we also saw how Truvada of PrEP changed the field in 2012. Once we start giving that to the at-risk population. You can see here in the graphic how many people got infected in big cities here in the United States and how much that decreased in 2017 because of Truvada. The big problem is implementation. It's not only about HIV, it's about a lot of other diseases. It's about pregnancy, unwanted pregnancy. We still have a way to go and learn how to make a better way to communicate with those communities and to make sure that the people are tested, people that are HIV positive, they are tested, or people that live with HIV, they are tested, and for people who are at risk of being infected, that they are protected with Truvada. So when I'm going to talk, what I'm going to talk here for bNAbs is just one more tool in this arsenal of weapons that we have here to control the epidemic. But it's not only the epidemic. We know that art improves the quality of life, but we also know that even with this control, there is still a low level of inflammation in those patients that can increase a number of other types of morbidities. They're not related to the classic AIDS, but can cause a lot of problems, especially during aging. You cannot forget that in Africa, we have a lot of children that were born with HIV and are being treated for a long period of time, are going through adolescence now, and at times they stop taking the drug and you have resistance. So even now, with all those weapons and all those tools that we have, we still have to think of ways to improve the quality of life of those patients. And why we're talking so much now about bNAbs. I agree with Norm that bNAbs are being kind of heralded now as the new tool and the new way of doing things. But why people brought up this? Don't forget that the idea of giving passive immunization to control pathogens is an old one. I mean, it is the beginning or the end of the 18th century that we had, 19th century that we have Paul Ehrlich, when he was developing a serum to treat diphtheria. And now we use ABS to treat a lot of stuff, cancers and inflammatory diseases. In fact, the 10 most advertised drugs in the United States, 2019, 3 of them were immunomodulatories, were like bNAbs. Some of them anti-human proteins like rituximab, which is anti-CD20, or anti-pathogens like rituximab for Clostridium. So the idea of using passive immunization for the prevention or control of HIV is not something new, it's not something so novel. It's just a question of bringing this from the past and put new techniques to understand how we can do it. In the past, we tried, in fact, to use plasma infusions to treat HIV patients, and we saw a decrease in, or transient decrease in plasma viremia, indicating that antibodies can be directly causing an effect in viremia and controlling infections. So not only T-cell response, but also antibodies. So why we're doing this? First of all, it started, I cannot say that it started because we want to have broadly neutralized antibodies. It started as a tool for the development of HIV vaccines. We want to have a vaccine that is going to generate one type or several types of antibodies that are going to be effective for the prevention of HIV. Studying this, what type of antibody we're going to need, that's when we found a lot of broadly neutralized antibodies, and then we start kind of focusing in isolating those antibodies and see what's the efficacy of those antibodies. Once we know what type of antibodies we need, we can start developing vaccines to guide the B cells for the production of those types of antibodies. And the VRC, that Gama scholar, who was the former director for the VRC, In fact, Rebecca Lynch is here, and she was involved in a lot of those research. They found that in the past, they analyzed several participants that had participated in trials that were living with HIV for a long period of time, including like elite controllers. And they would take plasma for those patients and analyze if the serum of those patients were able to neutralize a plethora of like variants of HIV that they had in the lab. And then they found this donor 45 that had a lot of, or a very nice profile of antibodies, an antibody that could really neutralize HIV or several strains of HIV. And that's when they isolated this antibody, doing like through cytometry and through cloning, and they got the VRC01 in 2010. This antibody has been tested a lot in several types of populations: adults, children, people live with HIV, suppressed, people live with HIV, viremic, for prevention. We have other much better antibodies now, and I'm going to present what we have in the field and how this can affect the CNS. So here are the HIV bNAbs in clinical trials, currently in clinical trials. You can see that this here is the spike of the virus, and we're highly glycosylated. So we have like few spots here where you can have space for antibodies to bind and to help preventing this virus from binding to other cells. So we have several of those sites. The most important of them, or the one that we discovered first, was the CD4 binding site. We have several of those antibodies already isolated and manufactured and tested in humans. VRC01, another variant VRC01LS, another one VRC07, 523LS, and 6LS, and now the VRC manufactured a new one that is more similar potency and breadth of VRC07, but might have a better half-life in plasma. It's called VRC123LS. Those in red were manufactured or developed by the VRC or DAIDS and during the NIH, inside the NIH. We have here in green those that were sponsored by Gates or acquired by Gilead, like 3BNC117, that now is used at Gilead for treatment, but we still can use this for prevention. We don't have to access Gilead, we can go straight to Gates. We have the other site, like the V3 glycan super site, the PTG121, and another variant, PGT121-414LS, and another one, 1074, all of them also in clinical trials. Another region is the V1, V2 glycan with Cap256, V2LS, and PGDM1400LS. And another area that we were so optimistic about, that is the GP41 AMPER, this area here between the spike and the cell membrane. The VRC developed a very nice antibody called 10E8VLS, and was extremely potent, extremely broad. And when we gave that to people, it was extremely, I cannot say toxic, but it gave a lot of adverse effects. And we gave that subcutaneously, and the antibody got trapped in subcutaneous area and causing panniculitis. So we had to stop this antibody and re-engineer or de-engineer this antibody to see if you could make it less harmful. I don't know where we are now with this research, but I'm pretty sure that we have people there at the VRC trying to make this this molecule a little bit better than we had before. But not only those sites, we have other antibodies now from companies that that are a little bit different from the normal antibodies that we have in mind. One of them is the Sanofi tri-specific. So imagine an antibody that has two FABs, and at the end of an FAB, we have another extension of that antibody that can bind to another site. We call that the tri-specific. So one part of the FAB is similar to the VRC01, the other one similar with PGTM1400, and the other one with 10E8V4, which is a different form of the 10E8VLS, and it's not harmful. It does not cause adverse effects. This tri-specific covers 3 sites, which is wonderful. So we can have less resistance when we give this drug. It is a phase 1 to now being tested by ACTG HVTN. We have another antibody that is bispecific. In one part, we have the 10A8V4 and one FAB, and the other FAB is the balizumab, anti-CD4 receptor. So this antibody was developed by David Hull's team and also and in combination with TaiMed, and they don't have a name. It's unfortunately don't have a name. We call the whole bispecific. I'm sure that this is gonna change soon. So those are the antibodies now being tested in humans. And we can kind of evaluate those antibodies by testing them in vitro in a panel with several viruses, several variants of HIV in several plates, and see how many of those variants, those antibodies can neutralize, in which potency or how much we need of those antibodies to neutralize in vitro. So here we have the C4 binding site antibodies, VRC01, 3BN, VRC07, and N6. We have here the V3, V1, V2, and AMPER. And here on the top, you can see like the percentage of resistant virus against that antibody. So VRC01, a very nice antibody, but it has like 30% of resistance in the population. And don't forget, this is a panel that has been done for a long time. It's a historical panel. So this does not reflect the current state of how many variants or how many viruses we have now in the population that are resistant or not to VRC01. It's very hard to do that. We have always like to be updating our panel. But here, when you see like VRC01 with this very nice potency, you can see that we have other antibodies that are even more potent, like VRC07 and N6, even with a much better breadth. We have antibodies that are potent with very high breadth, like AMPER, only 2% are resistant. And we have others that are very potent, but it is not very broad. For instance, Cap256, very potent antibody. The problem is it's very bad for clade B. It's very good for clade C and clade A. So it's a good antibody. In fact, it was isolated from a South African patient, and we're trying to use that in Africa, but it's an antibody we're not going to be able to use here in the Americas. So when you put this in a graphic to see the potency against breadth, you can see that all the antibodies, they are spread, very unevenly. So we have antibodies with S10E8, a big breadth and not a wonderful potency, and we have others that are very potent but not with good breadth. And our goal is to have like the golden slot area here with wonderful potency and 100% breadth. I don't know if we're going to be able to get that. What we're trying now is to develop some antibody combinations where we have 2 or 3 antibodies that in combination can cover a vast amount of variants. Here we have a heat map of a combination of VRC07, 523LS, PGT-N2N1, and PGDM-1400. And when you evaluate that and see, here you have in orange and red the viruses that are susceptible to that antibody, and in blue the ones that are not susceptible. So you can see the VRC07 covers a lot of antibodies, PGT-N2N1 not as much, and PGDM-1400 not as much. But when we combine them, we have at least one active antibody in that combination that covers 99% of the variants. We have around like 75% or even more of 2 antibodies active against those variants here. And the best one would have like all of them, sensitive to 3 active antibodies, but we're not going to be able to get that at this point. The big problem is when you see that with clades, we see, for instance, that some clades are going to be very hard to find antibodies to have a 3 antibody combination. For instance, the clade AE in Thailand. We still do not have a very nice V3 antibody to be used in this combination. So are there advantages of using monoclonal antibodies? A lot of people are very skeptical of why are you spending so much money trying to develop new antibodies or understand those antibodies. First of all, I'm very optimistic that this is going to be another tool for virological control. In fact, we have a trial now in ACTG, A5357, testing the combination of cabotegravir and VRC07 523LS as a double drug combo for the control of suppression. So you can suppress with a much stronger drugs and then transfer those patients to 2 drugs given every month or every 2 months because they have long life. We still do not have results from that trial. We know that it's very safe. but that can be used, for instance, instead of giving 3 drugs all the time to those patients that are suppressed, maybe giving 2 drugs every 2 or 3 months, or even every 6 months if you have a drug that can last that much. They are very long life. Some antibodies, VRC01-LS, can stay with high concentration in the blood for 6 months. It's better than use like a daily pill, or can be used ad hoc when you have, for instance, resistance against some kind of variant, and you can use that to complement that regimen. It is very low toxicity, and the 10E8V2-LS was an exception. All the other antibodies have very low toxicity, much less than chemicals. We haven't identified so far any anti-antibody ADA or anti-drug antibody. Even given like several administrations, like we was given like with VRC01, we didn't see any production of anti-antibody. Don't forget, those antibodies are human. They are not humanized. They are human antibodies. It is a wonderful tool for prevention in mother-to-child transmission. Imagine in Africa, a woman can come from her village, walk to the clinic in the little town. Once the baby is born, give 1 antibody or 2 antibodies to that baby that can cover 90% of strains. The mother can go back to her village without having to take with her pills or other drugs, and help that child to not be infected through breastfeeding. So that's one of the big goals of using those antibodies in places where you have a very high level of children that can be born with HIV. People say a lot about price. It is a very expensive manufacturing product, but it might be much lower than you expect. When you talk to some places in India and Brazil, they have already established very big vats that can be used like to produce those antibodies. And if the government or some commercial partners can help to develop those drugs, I think it's going to be not so expensive as people are thinking. Another thing that is very important is that it's a quick development and production. We know the mechanisms of antibodies. We know how they work. Think about like COVID, like even before the vaccine, we already had antibodies anti-COVID. Of course, the antibodies didn't cover all the variants. Omicron showed up. A lot of those antibodies stopped working, but it's still, we're able to isolate those antibodies very fast. Instead of trying to find a drug that binds to one of the enzymes and control the replication of that virus. And we have potential for improvement. We're going to talk about this later. We can increase breadth, potency, adapt the function of this antibody, and increase or change the form of administration to fit some types of populations, like children or people who cannot go to the clinic, for instance, every 6 months. And maybe the most interesting thing for you guys and for scientists is the possible immune modulation that this molecule can cause in patients or in participants that have HIV. Several papers were published in the past 2 or 3 years where they demonstrate that there is a possibility of a vaccinal effect when you give those antibodies very early during infection before the production of homologous antibody. So this is still being very controversial. A lot of people still don't understand what this vaccinal effect really is, if it is the formation of immunocomplexes that are acquired by the dendritic cells and presented in a much better form or a much efficient form. But you also saw a post-treatment suppression. So Michel Zweig and Rockefeller gave like 2 of his antibodies to people who are suppressed, people living with HIV suppressed, and they saw that in some of those participants, it took long, they stopped ART, and it took longer for those participants to have rebound, as if they were able to control the infection after giving the antibodies. So that's the vaccinal effect that people are talking so much. And other groups are trying to improve the antibodies for an increase in cytotoxicity. So antibodies, as you know, they can bind to infected cells, lead to NK and CD8s to find those cells and kill those cells. So that can be a very nice tool for cure or eradication of HIV. In fact, we have several trials at CTG. They're trying like cure protocols using bNAbs. Are they efficient? Are they efficacious? Well, let's talk about the AMP. AMP was the antibody-mediated prevention trial that the VRC did in collaboration with HBTN, HPTN, and DATES. It was a huge phase 2 trial. We had like two groups. One trial was done in the Americas for MSM transgenders, and the other group in Africa for women. We had more than 4,500 volunteers, and we finished this. So they received we split in three groups. One group received placebo, the other one received 10 milligrams per kilo of VRC01, and the other one 30 milligrams per kilo of VRC01 every 2 months for I think like 2 years. And this was published like some years ago. And if you say it was a success or not, well, it depends on who you ask. As a trial, it was extremely successful. It was a large-scale IV administration that we did in Africa, Brazil, Peru, and we were able to enroll more than 4,000 people with a retention of 95%. So think about that. PrEP, we didn't have this retention or the adherence when we did the PrEP trial. 95% of retention and adherence of 99%. In fact, 89% of people who we interviewed said that they preferred to have a drug be received every 2 months for prevention than having a drug taken every day. I personally do not agree. I think that I would miss, doses, but a lot of people would prefer to have this intermittent, receiving of a drug to prevent against HIV. Another positive thing is that this trial was able to give us a proof of concept that an antibody is able to prevent infection. The problem is the VRC01 was protected only against the acquisition of highly neutralization sensitive viruses. If you see here in that graph that I present to you, I showed you that 30% of that the viruses that from that panel, they are not neutralized by VRC01. If you consider that more of them or more variants are currently circulating in the population, and we didn't catch that with this panel, and consider they're going to need more than just like 1 microgram per ml to prevent infection. So of course, we didn't have a lot of viruses that were really susceptible to VRC01. The conclusion was that if you go to those panels in vitro, we see that those viruses that have the IC80 is smaller than 1 microgram per ml. They were able to be controlled by VRC01. For you to compare this in vitro result with an in vivo result, there are a lot of calculations. Peter Gilbert from Fred Hutch published a beautiful paper last year showing what levels of antibodies we're going to need in the plasma to have prevention. Huge levels, in fact. And we're trying to develop new, more potent antibodies because we believe that if we use the antibodies now, we're going to need a very large volume for the prevention of infection to get to that concentration in plasma. So what do I do at the VRC? So what I do is I control all those clinical trials that are using VRC, HIV, bNAbs. I did this graphic here for Julie Ledgerwood, who was one of our deputy directors for her to present in a retreat in 2019. As you can see here, the first antibodies or first trials started in 2013, and then cumulative. Here is when I started the VRC. You can see that after that, we had like a huge number of new trials. If you put the new graph here for 2023, you'd be kind of like here with more than 60 trials that are using or will use our antibodies or used our antibodies. Those trials were done in collaboration with MHRP, Caprice, HVTN, HPTN, IMPACT, IAVI, and ACTG, and a lot of independent PIs like Steve Dix, Dan Baruch, who used our antibodies for their protocols or regimens for cure or treatment. Here are all the completely trials that I had in my hands already tested like VRC or NIAID HIV bNAbs. So all those trials here, some of them were done in the VRC and all of them, a lot of them done in those networks or with our other collaborators. And here are those that are current being tested or in they have been proposed are going to start maybe next year. We have several of them here in green that are going to start next year. So as you can see, it's a very large number of trials. We still do not, we're still doing a lot of, for instance, phase one trials with VRC07 523LS, which is kind of like our flagship here for which are our best antibody or the one that are being tested more. And they still like testing that in collaboration, or not collaboration, in combination with other antibodies, or in the presence with latency reverse agents, or with other bispecifics, and see if that antibodies could be used in those regimens. So that's what I do for a living. I am in all those protocols. I have to evaluate all those protocols, being all the calls, make sure that everybody's using our drug correctly, make sure that we don't have a lot of adverse effects, and evaluating proposals that need those drugs, kind of like the gatekeeper for the antibodies. And when people, at times, I receive emails from, in the past, when Joe Mascola was the director, like, talk to Lucio, I'm not gonna deal with this, talk to him. So I'm kind of a diplomat of for all those drugs. But then I'm gonna talk here about ABs and the CNS. And I'm sorry, I'm gonna disappoint you guys, because we do not have a lot about ABs and the CNS. And maybe that's why it's so important for me to present that to you, because I want to hear from you guys what you think would be the use of bNAbs, or how we can use those drugs in your context. I work a lot with CNS, like in monkeys, as Norm said. I'm in contact with a lot of neurologists that are trying to use that, or to understand how those antibodies can affect the CNS. But it's a very hard proposition. So, for instance, the first question, can we detect bNAbs in the CSF in treated patients? There was only one PI that was able to show that in one study. When we try to pass that or ask other studies to add, for instance, lumbar puncture, it is very complicated. Most of the PIs don't want to do that, or increase the cost of the trial. They have to train much better, like the staff, and adds a lot of burden for those PIs. So the other question is, how can we improve the bNAbs to work better in the CNS? I'm going to talk a little bit about that later. And would bNAbs be useful in CNS disease or eradication? I'm not sure. I still do not know if that's going to be an important part for you guys. And that's why I want to hear from you what you think about it. That's a very new merging of two fields, which is bNAbs and CNS. So as I said, the only team that tested the presence or quantification of an antibody in the CSF was the RV397 trial in Thailand, where they gave VRC01 very early in infection. And then they took those patients from ART and saw how long you take for those virus to rebound. And they saw that those who took VRC01, it took a longer time to rebound compared to the ones who didn't take VRC01. So that was the only, in this trial, Serena Sputic was able to contact Gentanat, which was like the PI at that time, and she was able to obtain CSF from some patients, not a lot. She got in contact with me some years ago, and I convinced Adrian McDormand from our vaccine immunology program to test. And here are the results. We had one of our wonderful scientists, Madhu. She developed this test in the CSF. We didn't have this test developed specifically for the CSF. We had for sera, but not CSF. And what I want you to take from this is a little bit complicated table, but what I want you to take from this is here that the CSF before VRC01 infusion, yes, before VRC01 infusion, we cannot find any antibody there, which makes sense. But after, once the virus rebound, we saw a small amount of VRC01. In the picograms or nanograms at levels. In the serum, on the contrary, we see like before, of course, no antibody, but then after doing viral rebound, we have a very high concentration in the serum or plasma. But if you see here the date that was where the CSF was collected, it's like 16 days after infusion of VRC01 for this participant, 4 days, and for this participant, 2 days. And there's not a lot of correlation between the time that we collect the CSF, the concentration in the CSF, or even the concentration in the CSF and the concentration in the plasma. We do not know if there is always like a very strict control of transfer from the plasma to the CNS. We still do not know why we do not have this correlation. So I just have here like different types of presentations of this data. So here we have like CSF, the levels of BRCA1 after infusion, and here serum, you can see that it's like several logs of difference. So the conclusion from this very small study, and think about that, was just 3 samples they were able to get. We saw that the BRCA1 levels were like around like 3 logs higher in sera compared to CSF in the study. The CSF levels did not correlate with the plasma levels or the time of infusion. And we were going to have at least now to test like the IgG levels in the CSF to see if it was the same level with those that didn't receive BRCA1, or if this impacts the transfer of those, the traffic of that antibody to the CSF. And that's the only human data that we have. So we still don't know, for instance, how they cross the blood-brain barrier properly. We know that they can cross, and we could see that in that trial. We don't know which receptors are really involved in this transfer. One of the hypotheses is that the FC and neonatal receptor might be able to catch those antibodies from the plasma and then traffic them to the brain. I already read the opposite, that the FCN, the neonatal, is inside the brain and helps us to remove antibodies from the brain to the serum. So I still don't know exactly if people have a very nice understanding of this process, how the antibody can go or leave the brain. I know that we have a lot of antibodies now being evaluated for Parkinson's, for Alzheimer's, but not with a lot of success. And so I'm still waiting for you guys also to try to understand how this happens. And if Joe is here, we're in our lab, we are trying to develop a little model to understand also, give like macaques some SIV, anti-SIV bNAbs that we have in the VRC, and then analyze in the brain how or where those antibodies can be trafficking. How can we prove them then? So one thing that we did was we made some mutations in the FC part of the antibodies, and those mutations are called LS mutations. We have several of those mutations that can improve the half-life of plasma. And here is like there's 4 graphics for you to see VRCA1 in black, and we gave like several times VRCA1 to some participants, and the VRCA1 LS, the one that has the higher half-life. Look at 5 milligrams per kilo. You can see that for VRCA1, very rapidly goes under 10 micrograms per ml. But with 5 milligrams with VRCA1 LS, it lasts like much longer in terms of weeks. See, like 40 milligrams per kilo, even after like 20 weeks, we have more than 100 micrograms per ml of this antibody. There is one study, HBTN 116, that gave VRC01 and VRC01 LS to 2 groups of participants, and checked how much antibody they had in the plasma and how much antibody they had in the mucosal, in vaginal mucosal and rectal mucosal. And they presented that like some years ago. I don't think that they published yet that paper, but VRCA1 LS was present in the mucosa after 1 year of the infusion. So maybe LS helps the traffic from the plasma to the mucosa, to other tissues. We don't know. We have other scientists in San Francisco that labeled VRCA1 with some radio label and gave a very small amount to participants to see where this antibody goes and measured by radiation in PET scans. And he sees that VRCA1 and VRCA1 LS, they go to different places. So we still don't know how those antibodies really traffic in tissues or if they go to the brain. We don't know yet how the physiology of those antibodies, or what's the difference between LS and non-LS. And my question is, would it increase or decrease the concentration in the brain? In fact, the first thought that I had when I was discussing that with our team there at the Hopkins. I said, maybe we could give the antibodies to macaques, the version LS and the non-LS, and see how, compare to see how they fare in terms of CSF or tissues. There is no trial that is going to happen where we're going to have an antibody that is non-LS and LS given to people and see how much we have in the CSF. Why? Because all the antibodies now manufactured, they have the LS mutation. All of them. The goal was never to use the antibodies for treatment. The goal in the beginning of the VRC was for prevention. So the idea is, let's have molecules that stay longer in the plasma. So all of them now have LS mutations. I keep trying to convince Rick Kalp that we should go back and make some of those molecules without the LS mutations for instance, for cure regimens. Because one of the problems with cure is that we're going to have to do ATI to see the efficacy of your regimen. And if you still have like a tail of antibodies that are present in those patients after, the ATI, we can have resistance very quickly after, against those antibodies. So I'm sure that the FDA is going to ask for the measurement of very low amounts of antibodies once we have to stop ART. In fact, the FDA even asked the A5357 to measure the level of sensitivity to cabotegravir and VRC07 for all the participants in that study. Participants that did not have a result from that assay, which was a very complicated assay because those people are suppressed. They had to use a lot of PBMCs to isolate the DNA and see if they can find viruses there that are susceptible or not to those two drugs. A lot of those participants, maybe like more than 50%, were unable to be enrolled in that trial. So maybe with a non-LS, form, we can have like faster trials instead of have to wait 6 months for the antibody to go down and then do the ATI. Another way to improve, and I already told you, is to develop some antibody with bi or tri-specificity. We have already a lot of those antibodies being made for HIV, for even like for COVID, for other diseases. And there is one, I saw that in one paper where they're suggested to have a bispecific in where one FAB is going to be your antibody or a part against the virus, for instance, or against, I don't know, beta amyloid or another protein that you think should be, neutralized in the CNS. And the other FAB should bind to a receptor in the blood-brain barrier arm part outside in the blood. So for instance, the RMT receptor. So you can have an antibody that binds to the RMT receptor, and this antibody is trafficked to inside the brain, and then can be more efficacious. This hasn't been tested yet, but it's a very nice hypothesis that maybe we can have those antibodies going better inside the brain by modifying their FAB. The problem is, we don't like to fuss around with inflammation in the brain. So is that a good idea to increase the level of antibodies in the brain and increase the inflammation or no? Or maybe it is better to not have antibodies in the brain to prevent any type of complications there in the CNS. So we do not know, not even that. We haven't seen any problem or CNS complications in those patients that received our antibodies, but we did not have a lot of people seeing it. We didn't have a lot in those trials. We don't have a neurocognitive assessment. We don't have a lumbar puncture analysis. So we do not know. We really do not know how much those antibodies can interfere with the CNS. So first of all, we have no clinical or animal data yet. We start having that, but haven't been published yet. We know that the CTG is very committed. I'm in the bNAb work group with the CTG together with Katie Barr from University of Pennsylvania. We are very committed to address like the neurologist's concerns, trying to add neurologists to all the trials now that have bNAbs, amend the protocol so that, for instance, Leah Rubin's neurocognitive assessment can be used in those participants that have ATI. We are trying to convince some sites to do CSF evaluation, very tough. And we have a big pressure from the sites. They don't want to do that. It takes longer time. the participants have to stay like 3 hours already there. So if you have to add a neurocognitive plus CSF evaluation, it's going to just be more burden for those sites. So suggestions. I need to know from you guys, what do you think would be the best way? If it is important to understand how those bNAbs are going to interfere or not with neurology. And that's it. So I think that now we're going to wait for your question and your comments and start a dialogue to make sure that we can get to a point where we can at least like decide what we should do here. Should we really like spend more money and do like the macaque studies, or you guys think that maybe it's not going to be so important? Maybe that bNAbs are going to, maybe be worse. So I thank you, and I'd like to acknowledge everybody from the VRC. It's a very large group. The VRC is more like a biotech with production and R&D, and also to like all our collaborators and participants of the trials. Thank you.
Good. Wonderful. Thank you. Really informative and interesting talk. I'm going to forego, I put a question in there, but we'll get back to that if we have time. Let's start with Prasanta. Dash.
Very nice talk, Dr. Gama. And my question is, what do you think the effect of bNAbs, whether single or double, on the already existing latent reservoirs, including brain? And do you think other approaches like gene editing approaches, CRISPR-Cas9, in combination with bNAbs can have a better effect to eliminate the already existing latent reservoir?
Yes, I completely agree with you. But we do not have any data on that either. And we do not have people thinking about that. That's another problem. We need people that are going to start like developing some protocols, or at least some in vivo studies in animals that we can use. For instance, think about that. All those trials that we are doing now, not trials, the animal studies that we are doing, we do with SHIV. We have to do with SHIV because the bNAbs that we have are human bNAbs. So we have to have the HIV envelope in those, in those viruses. Mm-hmm. We have, now Mario Roeder developed some of bNAbs, there are simian bNAbs to be used against SIV. But all of us who work with macaques, we know that SHIV models are not very good for neurology. They do not go to the brain very well. I think the Katie Barr has a nice model now. I'm collaborating with her. But we have to start doing that in animals first and then pass that to humans, but nobody's doing it. We have a very few people doing that. It's a great question, but it's something that we're still going to need, some like in vivo, data to see how that's going to pan out in the future.
So can it be tested in humanized mice?
Humanized mice. We use humanized mice to check for PK in our antibodies. In fact, the humanized mice is a humanized FC receptor mouse. It has all the FC receptors from humans, including the FC neonatal receptor. It was the best best animal that we had to correlate with human PK, better than macaques, in fact. But we don't have anybody doing any type of brain studies with the mice at all.
Thank you.
And you had another question. Did you want to go with that one as well, along with gene editing approaches, the combination?
I combine both.
Okay.
Yeah. Thank you.
Arpan.
Hi, Dr. Gama, great presentation. And so in continuation with Dr. Das' question, actually, so do you have any data on the penetration of these bNAbs in the lymphoid tissues like gut and the lymph nodes, which are the major reservoir sites?
So yes, we have some kind of data in animals. In fact, Tom Hope from Northwest, he is doing some tagging of our antibodies with fluorescence and then giving to macaques and then analyzed where this antibody is going. He already has some data. He presented that in several studies, but haven't published yet. So we haven't done that yet, but we have groups that are trying to do it. Yeah.
Thank you.
Amanda has her hand raised.
Amanda.
Oh, wonderful. Oh, thank you, Lucio. It's just so wonderful to experience your energy and enthusiasm and wonderful work, and just wonderful to see. I had a couple questions. You mentioned that resistance to these antibodies can develop. And so I was wondering, is there screening of the applicants before? Because is this a phenotype of a swarm of viruses? So is there selection? So there's two parts, right? I think you
Yes, I
understand.
mean, that was what I was telling in the end, Amanda. We have a company called Monogram who developed I think Rebecca was part of the group that helped like with development of that assay. Katie Barr she was the one who developed a little trial in ACTG. So they have an assay where they take PBMCs from participants and then they isolate the DNA and do like pseudoviruses to see if those viruses in the reservoir, if they are resistant or not.
Okay.
Problem is those patients have been suppressed for such a long time that 60% of those patients we cannot isolate anything from them. So those patients cannot be enrolled for AP53, 57. And now there's a big controversy, Rebecca can participate in that we don't want to use Monogram anymore. We put so much effort there and now it's not like a useful technique. In fact, the VRC is trying to do like QVOAs to see if QVOAs are going to help with that.
Yeah.
And there's another problem. Is the reservoir in the blood?
We need a better assay. Yeah.
Okay, so
We
you
don't
mean
even
that they're
know.
defective? They can't even replicate?
Some of them are not. Yes, exactly. So we don't even know if they are in the blood. Maybe we're going to have to isolate from the tissue. But then imagine we're going to have to do biopsies from lymph nodes. I mean, it gets more and more complicated. And then when we talk about reservoir, then all hell breaks loose because it's a very small amount of genomes that you can acquire from those participants.
And the second one, that's more on the development side, but are there any thoughts about going intramuscular? So
Yes.
Yeah, okay.
Yes. We had one study, HVTN 127, where VRC-0753LS was given IV, intramuscular, and subcutaneous. I mean, it worked fine. Everything went fine. PK was fine. The problem is what I told you guys in terms of concentration. Our antibodies the concentration is 100 milligrams per mil. So you would have to use, I don't know, maybe 12 mils of antibody. I don't think we're going to be able to give, 12 injections or even like 6 injections, intramuscular. The goal is to try to go subcutaneous. I didn't say anything about it, but there is one company that developed a cloned hyaluronidase that we can give together with drugs. And once you give that subcutaneous, it kind of open more space in the subcutaneous region, and you can put more liquid there. You can use that, for instance, in children, where you can put saline. It can get up to 200 mil of liquid when you give together with hyaluronidase. Caprisa tested one of our antibodies with this enzyme and worked very well. So maybe in the future, we're going to have combinations of that enzyme together with antibodies to be able to use the subcutaneous area and give those antibodies. Problem, this enzyme is really expensive. They're charging us millions of dollars to try to co-formulate with our antibodies. So
Thank you.
And I think the name is Sid that has their hand up.
Yeah, thanks, Norman. Doctor, that's a great talk. Again, just going back into the same question, I think you've done so many studies now. The big question here is, what is the optimal concentration of antibodies required in the CNS in order to properly function or reduce the CNS reservoir? What is that level? Kind of, we struggle with that because whatever we give in the, intraperitoneal and how much gets into CNS, and if that is good enough or not, and based on your different versions of the antibodies you guys created and along the way, and what is your message? It's kind of somebody want to look at the CNS reservoir with any antibody for HIV cure. I think that's something I felt is very important for us to discuss here.
yes, I completely agree with you. And the answer is, I don't know. I have to confess, and Rebecca, help me here. We don't even know exactly what is the level of antibody they're going to need for prevention in the serum. We don't know that. we still don't know exactly what is the level of antibody they're going to need in the plasma of participants that are suppressed, and how much we have to keep them. 10 micrograms, 10 times the IC80, we do not know. We have some formula that we have. So for instance, Peter Gilbert did the one from AM study. Evan from the VRC did it with the RV97. And the levels are so high, like 50 micrograms per ml. I mean, we're going to have to give a lot of antibodies. Now, we have no data on CNS, nothing, zero.
Okay, thank you. Thank you for that. Yeah.
And Katie has a comment, but it's an important discussion point. So if you're available, there we are. Hi, Katie.
Hi. So first, I'll say I'm an early postdoc, so I may misunderstand some of this, but I guess I don't really understand how we can do bNAb studies without doing the CNS studies, especially both in CSF and brain. You know, we really don't know, and it's important to see both whether, you know, if it works systemically, say the bNAbs work systemically and control the virus, we still don't know if there can be active reactivation in the brain, and that may be replication competent, possibly. But even if it's not, even if there's reactivation of just transcripts of proteins, we know that that can drive significant neuroinflammation. And then on the other side, as you mentioned, we also don't know if the bNAbs themselves are going to cause neuroinflammation. So I guess I'm shocked that we would not do CSF at least.
Or brace yourself for more surprises in the future. You know, CNS and cardio are two parts of AIDS research that are not very well understood, and we don't have a lot of people that want to go there. Let me tell you how hard it was to publish showing that the brain had a reservoir. And I mean, Amanda, Norma, Avi, I don't know if Avi is here. It is very hard. For a long time, it was almost impossible to publish it. The backlash against CNS macrophages was huge. And now we still have people that say that it's not macrophages, there is T cells that are there, not macrophages. and I'm like, my God, you know, why are you trying to be so, you know, anti-field? Yeah. I completely agree with you. And that was one of the reasons that I started trying to push that for ACTG.
Yeah, I realize I am preaching to the choir.
Yes, I mean, I completely agree with you. And that was Scott Latundre and Tim Ma and Leah. We had a group, and now Rebecca Van Hughes from our group. We're going to try to have a grant where we're going to compare macaques with humans.
Oh, and yes, Scott's my mentor.
But let me tell you, it is part of the field. It is not about bNAbs. It is part of the field. Hard to get, hard to convince people. Tissue, very hard to study because we're doing humans. So it's an organ that you cannot just take a biopsy. So, you know, so it is really hard. I completely agree with you. I was also appalled when I heard for the first time. And I was more appalled afterwards when people say, why are we going to be doing that? It's unnecessary.
Yeah, what is the argument of why it's not? Do they?
Because after ART, we had a big decrease of HIV dementia or HIV-related dementia. We still have comorbidities, but if you're not a neurologist, you're not paying attention to that. That's one of the reasons that I want to have the neurocognitive assessment in all those trials. You don't have to have a CSF. You can just give like the iPad and see if the person is, you know, like better in terms of coordination than it was before doing when they were receiving drugs, for instance. That's not a big deal at all. But ask Leah how hard it was to push for that. I mean, it's amazing. They say that is not necessary. Oh, I haven't seen anything with my participants. And then when I talk to other PIs or other doctors from small clinics, for instance, in Baltimore, and they say, "Oh my God, I see all the time." You know, I see all the time.
Yeah.
You don't see
neurocognitive changes are going to be late. Sorry, go ahead.
I just said you don't see what you don't look for.
Exactly. But I agree with you 100%.
Yeah, Lucio, sorry, to continue that neurocognitive test, looking at every patient, the problem, most of them, it kind of, yes, it gives some data, but maybe not perfect as well, right? Because we miss baselines, we don't have same person, and then what we are looking is like a group, and then we're going to compare with what was not before. I think a lot of these are complicated in order to really make a real assessment is really going on just due to the drugs or HIV
But
or
Sid,
something else. Now, COVID is added another big factor.
but Sid, don't forget, we are doing that in a kind of like a clinical trial style. So we don't know how much of that is going to be able to be translated to the day by day. But those participants were trying like to have like the assessment while they are in art and after they are in art. Of course, we do not have pre-infection, but at least we know what's the baseline when they are completely suppressed. We know the amount of virus, the size of the reservoir by QVOAs, blah, 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 blah. And then we're going to do the ATI, and then we're going to see, did it get better? Is it still, you know, like it is still with the same type of results that we have baseline? At least that, but I completely agree with you. It is another complication. I know.
Yeah, thank you.
So I'll at Norm's prodding, I'll speak up about what I typed and just put another did you think about. You know, I know we're talking about neuro stuff here, but also, you know, relating to the idea that when we do these bNAb studies, you know, what you need to get protection is always these huge amounts, is that maybe we need to be really thinking about region specific penetration of whatever that antibody is. You know, to me, you know, mucosal surface for protection, maybe with a memory that, you know, the entire immune system has evolved to specifically give the right thing in the right place. And if we're just talking about IgG, that's a blood issue, right? And that maybe there's something else.
Yes.
Into the right tissue. So preaching to the choir again, but you know,
and you know, we are trying to develop like IgAs, for instance. We have groups that are already trying to develop IgAs, much more complicated because then how we're going to give that, correct? I mean, it's still like we have to understand the process of how the IgA gets there. And it's not just like, maybe for COVID, it would be like just spraying the nose, but for HIV, it's going to be very complicated. So I agree with you. We cannot make this correlation between what happens in blood and what's going to happen in the tissue. We can't. And but we're thinking of it. We are thinking of it. And maybe IgG is going to be able to. We saw that only with IgG we're able to control AVRCO1. So there is some hope there. Maybe it's not going to be the best, you know, with IgG, but at least we have like our foot there on the door.
Yeah, I'm not poo-pooing it. I guess I'm wishing you luck, and I'll watch from afar. Thanks.
Norm, you're mute. You're mute.
Oh, sorry about that. Thank you. I'm going to read a question from Tommy. His microphone is not muted, it's broken. And then we'll go to Jules, our community advocate. So Tommy asks, I am wondering if there's consideration to use focused ultrasound to make blood-brain barrier leaky in order to deliver antibodies.
First time I'm hearing. So I don't know how this, if it is possible to do that with animals. Do we have any data on that on animals? The use of ultrasound? I don't know.
I don't know. If you type something, I'll let you know. And I have the answer to that. That's an interesting question.
Yeah, it's a very interesting question, but we have no knowledge of that. And again, it goes to what Pam was saying, you know, and Katie were saying, that is that a good idea? You don't even know if it's that a good idea to increase the amount of antibodies there. So I think we're going to have to really test first if we have no problem about increasing the amount of antibodies in the brain before I think that we do something to really open the gates.
And then let's go to Jules from a patient standpoint.
Well, I think I think the whole field is really overly optimistic here about even the use or development of this. First of all, the resistance issue at this point in time seems to be a major problem. You can't really accurately identify resistance well enough. Number two, resistance seems to develop all too quickly and prevent success all too often, enough that it becomes a problem if you're going to use this in patients. Number three, infusions for long acting that's a real barrier for use for patients. And then lastly, this whole thing, there's no around the brain. I mean, it seems to me, unless you have to prove safety in the brain, otherwise you can't develop this. So with all these barriers, I just see over optimism about an over investment of money in all this when there are so many other areas that need attention. That's my comments.
Jules, I completely agree with you. I'm not disagreeing. I think that I cannot say there is like a over optimism, but it is one thing that I observed in science. When something gets tracked, it grows very fast. So for instance, we start Zika, all of us jumped into Zika and then put a lot of money in Zika and monkey model for Zika disappeared. Done, over. What's going to happen with COVID? I don't know. I'm going to keep doing vaccines and antibodies. I'm going to still do it like this, all we did for COVID. And the same thing happens with HIV. So, but don't forget that in terms of resistance, we had AZT as well that we know also got a lot of resistance. We had to start doing 2 or 3 drugs. And that's what I said to you, that maybe we're going to have more antibodies. But I agree with your point of view. I think that there is a lot of money being, going to bNAbs from a lot of companies, from a lot of studies. And I see the importance of that more for pediatric. I love the idea of giving that in pediatric in Africa, which is when I push a lot. I don't know if Debbie precise here, but Debbie and I will talk a lot about that. But in the other parts, like in treatment, how much we're going to give, I agree with you. But don't forget, that's my job. I'm not here like to promote the drug. If I do not have bNAbs, I'm sure that I still can do other things. But it is something that is very hard to go against because it is not something that it is toxic. It's not something that it is horrible. It's not something that it is really harming the patients. So at this point, the optimism, as much as it's high, it is still under the umbrella of safety and curiosity. We do not know a lot about it. We're still going to have to understand how bNAbs are produced in the body so that we can make better vaccines. So as the bNAb is being studied for vaccinal, strategies, then we're going to take like this tail and start doing other stuff with it, like prevention, etc. In terms of investment, let's invest in malaria. Let's invest in tuberculosis. Why are we investing in HIV? How many people have HIV in this country? Why? So it is not about that. It is how much we're going to try to understand the field. And yes, we're going to have a lot of waste of money here and there. And maybe this trend is going to go down, in the next years. But I don't see that. I see that bNAbs are being used now for so many things that even for bacteria, like we have that for Clostridium, people are trying to use that even for TB now. So I do not know where we're going to go for HIV. I am a little bit skeptical like you are, but I try to be a little bit more Pollyanna because, if I don't do that, I don't work and I'm going to be depressed on the top of everything that is happening in the world.
Thank you. And thank you, Jules. And thank you very much, especially for staying for 2 things. 1, jumping up your talk to do us a favor and fill this open slot. And 2, for a great talk and great discussion. Really enjoyed it. So thank you again, Lucio.
Thank you, Doc. Bye.
All right. We'll see you again.